humans and chimps and we say, yeah, 98 point whatever % divergent, any given member of two members of a given species would be like 99.9999 something identical.
Well, each of us would be slightly different.
Very slightly different.
The only identical genomes are two people that are identical twins.
That's right, and even they're not absolutely identical, even at the genomic level. Okay, is this going to let me do this? I want to skip ahead, but the recording, I think, is preventing me from doing it. I may have yet messed up all here. Oh well. Okay, I think we're back on. Okay, so in summary, if we looked at this as two competing hypotheses, the one hypothesis is common ancestry, sort of standard evolutionary biology. Organisms that we see in the present are descended from ancestral species based on speciation events, versus some version of design or special creation that is non-evolutionary in nature. Okay, so that's basically the basic options on the table. Okay, when we look at homology, it's exactly as we would predict based on common descent. Likewise for redundancy, synteny, and pseudogeny. What we see with synteny is very consistent with the recent divergence. Homology, we see exactly that these organisms have the same genes at the DNA level as well as at the amino acid level for the most part, and that redundancy, the DNA code is exactly what we would predict based on divergence. What you end up having to go with from a non-evolutionary perspective is every single time you need to make an appeal to some form of design constraint that gives the appearance of these things without in fact being accurate. So we'd have to say, well, the designer is somehow constrained to this appearance of descent, even though we know it's not true, but there's got to be some reason that we don't yet understand why these things have to look the way they do. And you have to do that every single time, at every level of data that we've looked at. Okay, so these lines of evidence are... Sorry?
Can you just sort of
say
that again and it didn't quite...
Sure. The question, sort of at sort of a simply speaking level, why do these genomes look like they do? Why do they have this appearance? They have all the hallmarks of what you would expect if they were in fact once the same genome. They have the same genes. Those same genes have the same underlying DNA code, even when they don't have to. They could have different DNA codes and still have the same protein codes. They have the genes basically in the same order. There are some rearrangements, but they're quite small. And they don't need to be in the same order, even to accomplish the basic biology of having our basic structure. We see in other species that you can have many different gene orders and still have the basic form of that type of organism. Yet what we see in humans and chimpanzees is almost identical in terms of their order. In terms of pseudogenes, in both genomes we see the remnants of genes that we no longer use. And moreover, we see those same defects in those same genes in both organisms. Those olfactory receptor genes like I was talking about, humans and chimpanzees don't use those genes anymore. They're mutated. They've got several mutations in them that prevent them from being used. Yet what we see when we look at those defective genes is exactly what we predict based on those two species having inherited those defects from a common ancestor. Does that cover it?
So the idea with those predictions, looking backwards, is that at one time they were
the same
exactly
species.
The same.
Exactly. Yeah. But for many of these pseudogenes, that those defects were already present in that species at that time and have been inherited unchanged from that original species. Well, why did they have those genes? Well, at an earlier stage, before that speciation event, those genes were inherited from yet another species. And if you go far enough back in time, you will eventually find the species that use that gene in its proper form,
So
as we see passed on to other organisms. Like if you look at dogs, for example, dogs still have most of these olfactory receptor genes functional. So, but they're not proposed to have shared an ancestor with humans until you go quite a ways back. So that common ancestor would have had functional olfactory receptors. Those two species went their separate ways. What eventually went on to become modern dogs has retained most of it, but it's been lost in the lineage that led to first the common ancestor of humans, chimpanzees, and gorillas, and then likewise on to us.
Can you explain the other side?
Sure.
What you're trying to say there?
Yeah, for sure. Okay. It's just a fancy way of saying, okay, if God really did create these two species independently from one another and they have never shared ancestry, then there is no need for us, there's no reason why we would expect to find these features in the genome.
Especially the last one.
Exactly, especially the last one.
Like,
So
why would you have genes
So
that
why
don't
would
work?
God make it? Why would God make us not by means of evolution, but make it look like it was by evolution? You know, that's basically what he's saying.
That's basically the general take-home message. And Terry's right. This last one, all of these at some level, and I've seen it done because I read a lot of anti-evolutionary literature. I've seen it done at almost every level to say, well, there's just some level of functionality we don't understand that requires that these things be this way. But this one is the one that they choke on. And essentially what they rely on at this level of argumentation with pseudogenes is to say they're not defective and they have a function. And that's generally the argument. Now, to a biologist who can look at the sequences and go, no. Even if you can show me that a pseudogene has some function, and a very rare number of pseudogenes actually have picked up new functions. They're sort of there in the genome, and occasionally they get co-opted into doing something else because they're kind of sitting around. Now, it's very easy to tell that it's been co-opted into a new function. Just as if you were walking across that field, you saw that building, you know what that thing used to do. It may well have a new function. It might be habitat for birds, it might be who knows what, right? It might be doing something. But the fact that it's doing something doesn't negate the obvious nature of it. It used to do something else, and it's quite plain. Moreover, suppose that you are not just looking at sort of walking across one field. Are you familiar with the type of novel called a counterfactual?